Welcome to the second Immunology tutorial. My name is Alex, and today we will examine the world of B cells and antibodies, otherwise known as humoral immunity. First, we will be going through the structure of antibodies, after which I will talk through the various functions of the antibodies. And finally, we will discuss the cells that produce antibodies and exactly how these B cells are activated. So let's look closely at the structure of an antibody. Here is one I prepared earlier. There are several ways you can break down the structure of an antibody. You can see there is a smaller chain bound to a larger chain, and that this arrangement is mirrored on the other side. This smaller chain is called the light chain, while the larger chain is called the heavy chain. You can also break down the antibody into its functional components. So in orange at the top, there is what's termed the variable region. Now this is important because it is this orange region that binds a specific antigen. In red is the constant region, which is responsible for the effector function of that particular antibody. It is this red part that will bind to different receptors on other immune cells and have a particular response, while the variable region with its antigen binding clefts will bind the specific antigen. Now there are other terms you will come across. You can divide the antibody into two halves, with the upper half called the Fab region, while the bottom half is called the Fc region. As you can see, the Fc region essentially corresponds to the constant region of the antibody. So this Fc region also refers to the part of the antibody that has the effector function. Additionally, the Fc region may be called the isotype of the antibody. So constant region, Fc region and isotype are almost synonymous terms and refer to the effector function of the antibody. Now antibodies differ in their Fc regions, and in total there are 5 immunoglobulin isotypes or classes, with each having a different function. They are IgG, IgM, IgD, IgE, and IgA. Let's now go through the different isotypes. Starting with IgG. IgG is important for several reasons. IgG is the most abundant antibody in serum, and notably it can cross the placenta from the mother to the fetus, providing protection from infections, or in certain instances even causing damage to the fetus, such as red blood cell antibodies. One of the important functions of IgG is opsonization. Now this is a Greek term that actually means prepared for eating. So what happens is if there's a bacteria present, an IgG specific for an antigen of that bacteria can bind. An immune cell, like a macrophage, has a receptor for the IgG. This is called an Fc gamma receptor. This binds to the constant region of the antibody. So you can see how the isotype of the antibody dictates what the immune response is to that antibody, via the various Fc receptors on immune cells. The macrophages will then be stimulated to phagocytose and destroy the bacteria. IgG is also involved in a process called antibody-dependent cell-mediated cytotoxicity, or ADCC. If you have a large cell that can't be phagocytosed, such as a host cell, and it is infected by a virus, IgG can bind to foreign components on the cell and stimulate natural killer cells via the Fc gamma receptor to kill this infected cell. The final function of IgG is to activate the complement cascade. If a bacterium invades and IgG binds the bacterium, complement can bind to the immunoglobulin and activate complement. This can be quite useful, particularly if a bacteria has a capsule that may prevent complement binding directly to it. Antibody stimulation of the complement cascade is known as the classical pathway. So that's IgG. It's very important and has many functions. Next we will look at IgM. I briefly mentioned that immunoglobulins can be membrane bound and act as B cell receptors. IgM is important because it is the B cell receptor on naive B cells. These are B cells that have not encountered their antigen yet and haven't been activated. So here's the B cell with IgM embedded in the membrane. I should also mention at this point that IgD can also be found in the B cell membrane as a B cell receptor, but there are few of these and are of unclear significance, so I won't be referring to IgD again. IgM is also very good at activating the complement cascade, and IgM is in fact better than IgG at this function. One of the reasons is that IgM exists as a pentamer, so you can imagine there are more sites for complement to bind and thereby activate. Moving on to IgE, IgE is the isotype that is important in allergies as well as parasitic responses, and facilitates this by binding to and sensitizing mast cells. Mast cells are found in tissues and express the Fc epsilon receptor. As it exists now, it cannot respond to an allergen, but if IgE comes along and the Fc region binds to the Fc epsilon receptor of the mast cell is now sensitized. So if an allergen comes along into contact with the mast cell, the allergen will bind to the variable region of the IgE, it will cross-link the antibodies, which will then transmit a signal to the mast cell to degranulate and release histamine, and this is the basis of allergy and anaphylaxis. Finally, we will discuss IgA. This is found in mucosal secretions like the gastrointestinal tract, and is involved in a process known as neutralization. What this means is if there is a toxin that must bind a receptor on a human host cell to exert its toxic effect, IgA can bind to the toxin via the variable region and stop its ability to bind cellular receptors and therefore cause damage. So that is IgG, IgM, IgE, and IgA, and we don't pay much attention to IgD. Let's move on to our third and final topic, B cell activation. Here we have a B cell just hanging out in a lymph node. As they are naive B cells, these are expressing IgM B cell receptors. A specific antigen now comes along from the peripheral tissue and drains via lymphatics to the lymph node and binds the specific B cell. Remember, one B cell will express only one type of B cell receptor that is specific to one type of antigen or sequence of amino acids. B cell activation is kind of similar to T cell activation in that usually one signal alone is not enough for full B cell activation, and in fact requires two signals. You have an antigen that binds the B cell receptor, but for most B cells, they also require T cell help. In lymph nodes, there are also T cells that hang out in the paracortex, so activated B cells will migrate towards this area via various chemokines or signals. Now, the CD4 positive T cell that is involved has already been activated by a dendritic cell bearing peptide antigen with MHC class II molecules. For further explanation of this particular process, please visit our other immunology tutorial. This activated T cell will also migrate towards the B cell. The process of T cell help for B cells has similarities with antigen presentation to T cells. The B cell expresses MHC class II molecules, which will present peptide antigens that were taken up by the B cell. This is presented to the T helper cell and recognized by the T cell receptor. Resembling the co-stimulation step of T cell activation, there is an important signal sent by a special molecule on T cell surfaces. It is called CD40 ligand. This is important because CD40 ligand is the basis of T cell help for B cells. This binds the receptor CD40 on B cells and sends a signal that leads to several consequences. The B cell will form a germinal center in which important processes are undertaken. The B cell proliferates, known as clonal expansion. It will also undergo isotype switching, and we should pause here to recall from our other video the various cytokines released by T helper cells. Remember that interleukin 12 induces T helper cells to differentiate into T helper 1 cells, which themselves produce interferon gamma, which will stimulate the B cell to isotype switch from IgM to IgG. If we had a Th2 cell, this would produce interleukin 4, which would cause isotype switching from IgM to IgE. Onto the other things that the B cell does in a germinal centre. It will undergo somatic hypermutation, which will lead to affinity maturation. Now, I will only briefly explain this. Recall the variable region of the antibody, which is the part that binds the antigen. Somatic hypermutation is where enzymes change the DNA structure of the genes encoding the variable region. So there are changes to the part that binds the antigen, which means they are changing the specificity of that antibody, such that it will either increase affinity, or binding to the antigen, or decrease affinity of that antibody. The antibodies, and therefore B cells, that have greater affinity will be selected for, resulting in a population of B cells that have better binding to the specific antigen. This overall process is called affinity maturation. The final thing that occurs in germinal centres are B cells turning into memory cells, which are long-lived cells that allow the immune response to respond to the same antigen or pathogen in the future with greater amounts of high affinity antibodies with greater speed. Now, this is the basis for most vaccines. To summarise B cell activation, signal 1 is the antigen binding to the B cell receptor, while signal 2 is CD40 ligand on T cells binding CD40 on B cells, providing T cell help for B cells, and through this, the efficacy of the humoral response is improved. That concludes the tutorial. Thank you for joining me, and if you enjoyed this video, please check out some of our other tutorials on our channel.